Chapter 12, Complex Inheritance. Learning objectives for this chapter. By the end of this, you should know what determines biological sex, understand how X-linked or sex-linked traits are passed down, know how to interpret a pedigree, explain how Y chromosome analysis works, and know how incomplete dominance can cause a different phenotype. We'll also talk about things like polygenic traits, multifactorial traits, and codominance. Okay. So last chapter, in chapter 11, we talked about what we like to call simple inheritance, which is also equal to Mendelian genetics. Okay. I would highly recommend reading over milestone 4 in your textbook. This goes over Mendel and what he was doing essentially. So Mendel was this guy, he was actually a monk and was somewhat curious about genetics, and is kind of like the unlikely father of genetics because of it. But he basically just started crossing pea plants. So he would cross these pea plants and he would observe different phenotypes. So he could look at the seed color, the texture, the color of the flowers, etc. So he would be able to observe these things relatively easily. And because of that, came up with a couple different laws. So we have the law of segregation as well as independent assortment, which we'll go over here in just a second. But the law of segregation just states that when an organism produces gametes, or their sex cells, the two alleles for any given trait separate so that each gamete only receives one allele. So you're actually kind of looking at the second part of a Punnett square right here, but to get to this point, he actually crossed a purple flower with a white flower. We'll call it a true bred, right? So we'll say that the white was RR, the purple little r, and the purple was capital R, so dominant, right? And when he crossed these, right? So I can write it like this so it's a little bit easier. So if we cross them, we actually get all heterozygotes. Okay, so if we have a homozygous recessive and we're crossing it with a homozygous dominant, all of our offspring, so 100% of our offspring are going to be heterozygotes. Okay, so they have one dominant and one recessive. So the dominant one is going to show, right? So all of our flowers are going to look purple, even though they will be carriers for the white. Okay. We call these offspring F1. Okay, so F1 just refers to the generation, basically. So F1 is the first generation. So we have our generation here, it's the offspring of the first generation. So F1 would be the first offspring. And then this generation here, we would call F2. Okay, so these are the potential offspring for crossing these F1 parents. Okay, and you can keep going, F1, F2, F a million. You can keep going. It's just generations. So if I ask you to do something like that, it's just asking you to do a cross. So if I ask you to cross a homozygous recessive with a homozygous dominant, you can automatically tell me without even doing a Punnett square that the F1 offspring, the offspring of that homozygous dominant and homozygous recessive parent, are 100% heterozygotes. Okay, they have one of each allele, right? And then you can also pretty much automatically tell me if I take that F1 offspring and I cross one from each, I know I have 100% heterozygotes, so basically if I'm crossing two heterozygotes, here are my odds. So I have a 25% chance of having a homozygous dominant, a 50% chance of a heterozygote, and a 25% chance of a homozygous recessive. Okay, so those are my genotypes again, right? If I want to look at my phenotype ratios, I have a 75% chance of purple, okay, and a 25% chance of white. Okay. And you'll see here we have purple and white, but they're both represented by the same letter. So keep this in mind, especially when you do exercises, which I think you'll probably do in lab coming up, that you always want to use the same letter. If you use different letters, it gets really complicated. So I know that R doesn't really necessarily represent white or purple, but it'll get a lot less complicated than if you use W and P, because then you don't really quite understand where the dominance lies, right? So be careful with that. Always choose the same letter and then just represent it as a dominant or an uppercase letter and a recessive or a lowercase letter. And if it's hard to tell the difference between the letters, you can underline the recessive character. So whatever, like A and A, or even a better example is P's. So we can underline the recessive or the lowercase one. Okay. So that's Mendel's first law, is segregation. So again, two alleles separate, so each gamete only receives one. So we have our F1 parent. Let's say that this is a dad, even though we're talking about flowers. He has two alleles for this gene that we're passing on, right? So this is only one gene that we're talking about. It's not like every code of instructions that he's passing on to his offspring. We're just talking about one gene here. So he can either pass on a big R or he can pass on a little r. Okay, so there's only one that's going to be in that gamete, in that sex cell. Okay. The next law is independent assortment. So for this one, it just says that two alleles for any given trait will segregate independently from any other alleles when passed on to gametes. So this basically means that just because mom or dad has a certain phenotype doesn't mean that they're going to pass that on. So you'll see here we have a smooth yellow parent. So we have heterozygotes basically being crossed again. So we can kind of put them in the same situation. Let's pretend like we had a homozygous dominant and a homozygous recessive mom and dad, and then they created this F1 generation here. And then we're going to cross this F1 to get our F2 in this Punnett square. And keep in mind, this isn't your F2 necessarily. These are the possibilities or the probabilities of your F2 generation, but it depends on how many kids you actually had, right? And you might have 20 kids and they all have this one. Okay, so this is just the probabilities here. But if we have a smooth yellow parent, they're independently assorted. These alleles aren't necessarily always together. So even though you have capital R and capital Y, let's say, so the capital indicates smooth and the capital indicates yellow, but it doesn't mean that you always pass that on. So you might pass on a capital R and a lowercase y, which would indicate that you would be smooth and green, right? So you're not always passing on the exact same thing, which is how your kids look different than you and how you look different than your brothers and sisters, etc. Okay, so what happens when neither of these laws apply? This is what we call non-Mendelian genetics, or in our case, I'll also refer to it as complex inheritance. Okay. So when we go over complex inheritance or non-Mendelian genetics, that includes things like sex determination, sex-linked inheritance, incomplete dominance, codominance, continuous variation, multifactorial inheritance, and non-disjunction. Okay, and we'll go over what each of these are now as well. Okay, sex determination. And please keep in mind when we're talking about sex determination and biological sex, we're strictly talking about biological sex. We're not talking about gender. A lot of things can go into gender, and it's coming out more and more with more research, but we are strictly talking about biological sex. Okay. So we have sex hormones that are produced by our gonads. If you're a female, you have ovaries. If you're a male, you have testes. Okay. And we produce either androgens like testosterone, or we can also produce estrogen. Right? Males and females obviously produce both, but males, you produce a higher level of testosterone. Females, you produce a higher level of estrogen. Again, you do have both, and we want these in check depending on which sex you are. You might have more or less of one or the other, but you do have both of these. So in a fetus, your sex hormones are shaping the development of your sexual anatomy. And the way that your sexual anatomy is being developed depends on these sex hormones, which depends on the set of chromosomes that you receive from your parents or that the fetus receives from the parents. As you probably already know, your 23rd pair of chromosomes in humans is what determines your biological sex. So females, you either have XX, males, you have XY. Again, we are diploid organisms. So we have a pair of chromosomes here, but our whole goal going through meiosis is to become haploid. So females, you can either have, well, sorry, you can't either have, you only have an X. So the only thing you can possibly pass on to your offspring is an X. Males, in your sperm, you can either have an X or a Y. So you guys are the determinants of what sex the baby will be. Okay. So again, females, you're always going to pass down an X. Doesn't matter who it's to. And if the male passes down an X, then we create a female offspring. If it passes down a Y, we create a male offspring. Okay, so again, the Y chromosome decides. And this is a pretty cool picture of chromosomes. This is under obviously a microscope, and there's different kinds of microscopy that you can do. But this very clearly shows how much larger the X chromosome is. So the X chromosome is much larger. It carries a lot more genes on it. And the Y chromosome is much smaller. Also, something to note, the Y chromosome does not go through recombination in meiosis like the X chromosome does. Okay, so it doesn't have the ability to go through recombination and kind of change itself out, which means that as the Y chromosome is passed down, it's relatively unchanged. Okay, so boys, your Y chromosome looks basically the same as your dad's and your grandfather on your dad's side and his father, et cetera, et cetera. So it's relatively unchanged. Okay, so what determines biological sex? Again, our X and Y chromosomes are what determine our biological sex. There is a gene on the Y chromosome called the SARI gene or the SRY gene, and that is what specifically signals the testes to develop. Testes produce testosterone, and that is what is helping develop the sexual anatomy or the penis in males. There are lots of XY chromosome disorders, and your book is pretty interesting. It goes through an example, I forget, I think her name is Brenda, and she was actually born a male. I think they actually had a circumcision incident or accident, and decided to reassign her as a female. And as you can imagine, this created some issues. So they raised her as a female. They always told her she was a female, but she never felt like a female. And again, there's a lot of different, we're not getting into any sort of gender norms or talk of gender because there's transgender. There's lots of other things that come along with this. This is strictly biological that we're talking about right now. So Brenda was raised as a girl. Even though she was born as a male, she was just reassigned after birth and eventually figured out that she was a male and kind of went through the process to convert back, essentially. So this to scientists was kind of a big deal because we always, it's kind of a nature versus nurture debate. And obviously both play a significant role, and you'll see different research arguing for different sides of it, right? But this was a pretty big case study for the medical world as well about being pretty indicative that biological sex is actually very much shaping your sexual identity as a human. Um, and there's other chromosome disorders that you can have as well. And your book talks about intersex individuals like hermaphrodites, which we'll talk about here in just a second. They used to be called hermaphrodites. Now the more correct term is intersex usually. But the process for dealing with those individuals at birth has drastically changed. Okay. So XY chromosome disorders, there's quite a few of them, but they're just genetic conditions that can be caused or affected by the loss or damage of the sex chromosomes. So this can be a duplication, a deletion, etc. of chromosomes, which a lot of times, or not a lot of times, but basically what we call this is aneuploidy. Let me write this out. So aneuploidy oops, sorry. Aneuploidy. Aneuploidy is extra or deleted chromosomes. Okay, so for example, Down syndrome, right? What's Down syndrome? Down syndrome is an extra chromosome. Specifically, I think they have an extra copy of chromosome 21, which sometimes we call trisomy if they have an extra copy of a certain chromosome. But excuse me, aneuploidy is the general term for having either an extra or a deleted chromosome. Okay, and aneuploidy is caused by something called non-disjunction. I'll write it out here. Okay, and non-disjunction, just basically the failure of chromosomes to separate accurately during cell division. So aneuploidy is a direct result of non-disjunction. They're not separating accurately, and therefore you're either adding or subtracting a chromosome. So this is something that happens in, say, something like Downs. And Down syndrome is much more prevalent in females that are giving birth at an older age, because remember, our sex chromosomes, I'm sorry, our gametes. So for females, our eggs are as old as we are, right? You're born with a number of eggs that you're always going to have. You're not growing more. But basically, your eggs are stuck in the middle of meiosis 1, until you hit puberty, and then they're going through the whole process to actually become an egg and be able to be fertilized, blah, 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 right? So because of that, you're kind of subject, as they get older, you're subject to more um, failures and more room for error, essentially. And specifically when we're talking about non-disjunction, it's just, again, the failure of chromosomes to separate. This can occur during meiosis 2 of either sperm or egg cells. So I'll write this out here too. Meiosis 2. Okay, so this is the very end, whether they're separating accurately or not. This non-disjunction can lead to aneuploidy. Okay. So when this happens, you might have something like Down syndrome. You could have, there's a few other things that can happen, but one of the things that we're talking about with these chromosome disorders is a lot of times the phenotype does not match your chromosomal sex. Okay, and again, there's lots of different things that go into biological sex. There's also lots of things that go into gender, and those two are different. But with biological sex, there are genetic, hormonal, anatomical, and behavioral aspects to sex, right? And you can see that in your own offspring. If you have your own offspring, you can see that interacting with the world around you. You can see that in yourself, right? But again, females, you have XX, males, XY. Um, you can still have XX or XY and have pseudo hermaphroditism. And a lot of times what that means basically is that you're you have the wrong parts according to your biological sex, or it's fairly ambiguous. So you might have a really small penis or a really large clitoris, just depending on which sex you actually are. And again, a lot of times what doctors used to do is they would just reassign you at birth, sometimes with the guidance of your parents, sometimes without telling your parents. And that's led to a lot of issues as children age. And a lot of times now what they do is obviously consult the parents, but a lot of times they wait to make that decision until a little bit later in life, when you can kind of see more of the anatomical and behavioral aspects to the sex coming into play, especially the behavioral aspects. So they can kind of see whether it's you see more male or female. There's true gonadal intersex, so this is basically a true hermaphrodite, so XX and or XY, and this is where you have both ovaries and testes, so you've got a little bit of both, and again, these are the historical true hermaphrodites, also infertile. You have triple X syndrome, so now we're getting into some of the extra ones as well. This is where you would have ovaries and you are a female, you are fertile, so you can still reproduce offspring, but a lot of times it's associated with pretty heavy learning disabilities. You also tend to be taller than average. There's also Klinefelter syndrome, so an extra X here as well, but now you also have the Y, so you are testes, you do have testes, you are a male. you are infertile, and you do have enlarged breast tissue most of the time as well. 47 or XYY syndrome. So we've got an extra Y here. Again, you still have testes, still are male, and you are fertile, again, taller than average and at much greater risk for learning and emotional disabilities. And then Turner syndrome is where you just have one X. You are a female in the biological sense, and again, infertile, broad chest, web neck, comes along with some other kind of abnormalities. You do always have to have at least an X chromosome, otherwise you will not make it. Okay, so let's get into sex-linked inheritance. So we do have genes located on either of the sex chromosomes. So daughters and sons don't share the same probability of inheriting. And a lot of times there are Y-linked traits, but again, there are many more genes on the X chromosome. So you'll see, especially in this class, we'll talk quite a bit about X-linked traits. Okay, so keep this in mind. Whenever we talk about X-linked traits, it has to be associated with an X. It has to be sex-linked. So we did simple inheritance before, right? And we would just write out capital A, lowercase a, capital A, capital A. And this is what our inheritance was. This was our genotype. But now we're getting into X-linked stuff. So now a female would be XX, a male would be XY, and whatever gene they're carrying would come on that. So let's say I had my heterozygote up here, I would have an XA, capital A, and an X lowercase a. Oops. And then usually we don't put genes on the, or alleles on the Y. So let's say that I had a dominant male here, I would have an X capital A. So make sure when you're doing complex inheritance, it's always written like this. If you're doing simple or Mendelian genetics, this is how you write your genotypes. So they are very different and you'll get practice with this, but keep this in mind. If you do the sex linked for a regular problem, that's just simple inheritance or simple dominance, then you will get it incorrect, okay? So what's an X-linked trait and why does our probability differ between males and females? So X-linked trait is a phenotype that is determined by an allele on the X chromosome. Makes sense, right? Most of the time when we're talking about these, they are recessive. Okay, so these are the lowercase ones, which means that a normal copy on one of the X chromosomes can mask the recessive disease allele on the other X chromosome. So think about this. Females, you've got an XX. Males, you've got an XY. So if mom has the disease, the chances of a female getting it are 50%. Well, roughly, not factoring in dad. The chances that mom is going to pass it on to her daughter are 50%. Whereas the male, it's 100% because the only X he's getting is from mom. Okay, so this is one of the reasons why we see a different probability for males and females is because females, you still have another X, you have another chance from dad, okay, that you can mask that recessive allele. So one example of this is Duchenne muscular dystrophy, or DMD. Another example I like to use, and you kind of heard me talk about it at the end of last lecture accidentally, is color blindness. Okay. I'm not sure if any of you are colorblind. I am not, but my brother actually is, and no one else in my family is. So you see this much more commonly in males. Females can be colorblind, but it's much more common in males because it is an X-linked trait. Okay. So again, because a male has a single X chromosome, he shows the effects of any recessive alleles located on his X chromosome. So say we have a mom with a DMD allele. So she's normal, she's unaffected, but she is a carrier, okay? So she still has that DMD allele, so she has a chance to pass it on, whereas dad is unaffected, doesn't have a chance to pass it on, okay? So, and there should be something on here. So let's do like capital D, lowercase d, capital D, okay? So we have X, and you just have to pretend like this goes on here because you still need the X's with it. and then you'd have x dy, and then x, d y. Okay, and then x d x d. Okay, so we've got, and this is where sex-linked can get a little bit complicated, because now we can look at our offspring and say, okay, we've got a 75% chance of having an unaffected child and a 25% chance of having an affected child. But if you want to get even more specific, what's the probability of having a daughter that is affected? So the probability for this one, right, of having a daughter that's affected is 0%. You will not have an affected daughter because dad would have to have that recessive allele in order to pass it on. Okay. But you have a 50% chance of having a carrier daughter. I'm just going to abbreviate things a little bit weird. Okay. What are the chances that you have an affected son? Well, you've got a 50% chance you'll have an affected son and a 50% chance that you'll have an unaffected son. Okay. So sons, you kind of get the root of all of this because you've got a much greater chance of getting anything recessive on the X's from your mom, whether she's a carrier or she's affected, because it's going right to you, right? You've got a 50% chance of getting one of those, whatever allele she can pass down to you. Okay, so saying this, we have a chance of having a carrier mom. Can a male be a carrier? Okay, can a male be a carrier for an X-linked trait? That's what I should ask. No. Male cannot be a carrier for an X-linked trait. They are either affected or unaffected. Okay. So they either have a dominant or they have a recessive allele. If they're recessive, they're affected. If they're dominant, they are not. So they can never be a carrier. So as a male, you'll never be uncertain for a recessive disease, a recessive sex-linked disease, if you have it or not, because you will know for sure. Females, you're a little bit more difficult, right? You might be a carrier and you might be able to potentially pass that on to your offspring. So similarly now, if we go over and we do our little Punnett square to see what our offspring could potentially produce, we've got a father now who is affected and has the DMD allele. So he's X lowercase d y, and then we have an unaffected mother, and she's not a carrier now, so she's X capital D capital T. Okay. The odds of having an affected daughter, still 0%. Right? Because we would have to have a recessive allele coming from mom, but we only have dominants, so mom is going to save daughter here. Okay? So no chance that we're going to have an affected daughter. Also no chance that we're going to have an affected son because we cannot possibly pass down a recessive X-linked trait if mom doesn't have it. Okay? So no affected children here. However, your children, your daughter will 100% chance of being a carrier. Okay, so this is something to keep in mind. This is why it's really good to know a little bit of your genetic history. Say you know that you have this disease, it's nice to be able to know what you could potentially be passing on to your offspring and how that could affect further generations as well. So that would be your F1 offspring, how it could affect your F2 offspring, so like your grandkids, right? Because now, say your daughter, she's a carrier, she could potentially give that to your grandchildren, your F2 generation. Okay. And I didn't mention it in the last slide or in the last few slides, but when we're talking about some of these chromosome abnormalities like Down syndrome, the aneuploidy, one of the things that you can do when you're pregnant is you can go through something called amniocentesis. Sorry, I know this is backtracking a little bit. Amniocentesis. Oops, this is meant to be an I. Sorry. Amniocentesis. And that's basically where they take this really long needle into your uterus for females when you're pregnant to detect fetal abnormalities by karyotyping. Okay, So amniocentesis you can do when you're pregnant and you are looking for the karyotype, which is basically just what your chromosomes look like, what the number of chromosomes are. So this is kind of a way when you're pregnant for you to know if you are going to have a quote unquote normal baby. Okay, so this is an option that you can do to kind of help you prepare for the child that you will be having. Okay, back to sex-linked inheritance, which we often call X-linked inheritance. Again, mom's passing on her X chromosomes to children. Father is only passing on an X chromosome to the daughters and only a Y chromosome to his sons. Boys, you only have an X chromosome, so you have a higher probability of inheriting X-linked diseases than girls do. Okay. And to do sex-linked inheritance, so when you're looking at like family trees and stuff a lot of the time as well, we're using something called a pedigree to follow these inheritance patterns. So we do Punnett squares, right, to kind of understand. We can also understand sex-linked inheritance through Punnett squares, but it's not so much of a family tree. It's kind of to understand what your offspring could potentially look like. But pedigrees are a visual representation of the occurrence of phenotypes across generations. So usually males are presented by a box, females are represented by a circle, and then usually the lines in between are marriages, right? And then you have offsprings usually going vertical down. So grandpa up here, married grandma up here. And keep in mind in this example, instead of using letters on the X's, they're coloring it, right? So we're still going through DMD. So for example, for an unaffected grandfather, you could put X capital D Y, and we have a carrier grandmother here, so we have X capital D, X lowercase d, right? And you can keep going down. So you can see that, let's see, are any of them, this is the only one, yeah, that's the only one that's affected, right, is this male down here. Um, and you'll see in, I think, your class activity example as well, what you might have to do is work your way backwards to figure out who could have possibly been a carrier or what the genotypes were of different generations, okay? So like say we started down here and we know we have a male who has DMD. So his genotype would be X lowercase d Y. And he has a completely normal sister. She has X capital D, X, we don't actually know, right? I mean, technically we know because we're looking at it, but if we just knew it was a normal sister, she could potentially be a carrier. So we would have to know a little bit about mom or dad, but we would know that dad's normal because we would know if he wasn't, right? And we know mom's normal as well. So she's got to at least have one capital D. But in order for that cousin, the male, to have this disease, there must be a recessive allele somewhere. And he's only getting X from mom, so we know that mom has to at least be a carrier. Okay, and if we know that mom's at least a carrier and dad is a an unaffected individual, then we can kind of do what the probability is for this female. So we know she's at least got one capital D. There's a 50% chance that she could be a carrier. So at this point, we just kind of have to guess. But you can go backwards and kind of figure out what your pedigree would look like. Okay, you do have to have some information, but not necessarily all of it. Okay, so a little bit of fun with Y chromosomes and analysis. Again, they are very small and they contain very few genes. So we can study ancestry and identify paternity as well. So if in the case of not knowing who the father is for paternity, it's really easy because these Y chromosomes usually are not going through recombination or independent assortment. And because of that, your Y is essentially unchanged for generations. So for example, if a male is a suspect in a crime and you know who his grandfather is, or his brother, his uncle, any male related to him, okay, on his dad's side, on the paternal side, you can test those people, and if that DNA matches, you know that it's pretty much has to be that person, or it has to be a male in that family. Okay. So the example your book talks about is the Jeffersons. Okay, so sons inherit this Y chromosome from their fathers, and you can compare the DNA sequences because they're pretty much unchanged. Again, they're not really going through any sort of recombination here. So we're comparing these STRs on the Y chromosome. So the example that we have is the Thomas Jefferson pedigree. And I don't know if you've heard, there's quite a bit of controversy around Thomas Jefferson and whether he fathered children with one of his slaves. It was always rumored, and it was pretty much dismissed until a little bit later when scientists were like, we can actually use DNA to figure this out. So they did. So they analyzed the Y chromosome of descendants from Sally Hemings' family, who was the slave, as well as the Jefferson family, and basically found out that, yeah, they had the same, pretty much the same Y chromosome. The STRs on the Y chromosome were the same, which doesn't prove that it was Thomas Jefferson necessarily. It does prove that it was a male in Jefferson's family. So it's possible that Thomas Jefferson's brother could have fathered these children with Sally Hemings. But with the dates and times and everything else going on, they've pretty much said, like, yeah, for sure this was Thomas Jefferson. Okay. So that's pretty interesting and pretty cool to be able to do with the Y chromosome and with males. Okay, getting into a little bit more with this sex-linked inheritance, with the complex inheritance, we also have, again, it's just getting more complex. So we have incomplete dominance. So this is where there's two versions of the gene. Okay, so say we're talking about hair texture, right? There's only two versions of the gene, but there's three different phenotypes. Okay, so you can have curly, straight, and wavy. So let's say that we do W and lowercase w, right? So let's say we have a wavy-haired dad, okay, who would have, oh, sorry, I don't want, okay, let's say we have a straight-haired dad. So lowercase w, and then we have a wavy-haired mom. All of their offspring would be heterozygotes and would therefore have, oh, sorry, okay, we have a curly mom and straight-haired dad. Okay, and then all the offspring would be heterozygotes, so they would actually be wavy. This is what incomplete dominance is, okay? So it's basically where the heterozygotes have an intermediate between the homozygous dominant and the homozygous recessive, okay? So here's what I was trying to do. Curly-haired father mates with a straight-haired mother, and they have a 100% chance of having wavy-haired offspring. And obviously genetics sort of sometimes stump us. Sometimes this doesn't completely hold true. But this is what incomplete dominance is. It's just basically a nice intermediate. So I try to remember it: incomplete, intermediate. Okay. Pretty straightforward. There are some other examples as well, but intermediate between the two homozygotes. We also have something called codominance, and the thing that you'll see this most associated with is blood. Okay. So with codominance now, we have both alleles contributing equally to the phenotype. Okay, and when you're thinking about this, think about what your blood type is, and you might not know your blood type. I actually used to do this in lab. I would make the students blood type themselves, which is really cool, but freaked a lot of students out because you had to use one of those quick needle things. But we have different blood types, right? And we have 3 different alleles for blood type. But think about when you're getting a organ transplant or a blood transfusion. That transfused blood, or that organ transplant even, they have to be compatible in ways that are determined by your genetics. So you can't just give blood to anyone. Your body will reject that. Okay. Both maternal and paternal alleles are contributing equally and separately to the phenotype. So this is a little bit different because we've got 3 blood group alleles. We have A, B, and O, and these are our possible genotypes. Okay. Again, they're codominant, so we can have OO, AO, BO, AB, AA, and BB. So blood types for A and B, those alleles are codominant. O is recessive to both A and B. So if you have AO or BO, that just means that you have either A or B. So you basically just throw the O out there. The only time you will say that you have O type blood is if you have an OO. Okay. And these are signified by what kind of markers you have on your blood. So if you have type A blood, you have type A markers. So this would either be an AA or an AO. Type B markers here, you either have type BB or BL. Okay. And it says who you can donate to as well. You can also have both type A and type B markers, which would mean that you are an AB. Okay, and then you could also be a blood type O. So the universal blood donor is O. It's actually O negative, which we'll get to the positive negative part of it in a second. The universal recipient, so this is donor, O negative. The recipient, which is ideally who you want to be, right? You want to be the person who can take blood from anybody, is AB positive. Okay. So that's what codominance means. And again, the biggest example you'll see of this is blood types. So if you have AB blood, you have both A and B. O is recessive. So you either have O or you have A or B that also has an O with it. So with our blood types, we also have something called RH, and it can be either positive or negative. The positive or negative just means that you either have presence or absence of these Rh proteins on the surface of your red blood cells. Okay, so like a type O negative, which is our universal donor, right? They do not have surface markers that can be recognized and rejected by a recipient's immune system. If you do have positive surface markers or you're something positive, then it means that you can be rejected by other different types. Okay. So again, this is dominant or recessive. You can also be a positive donor. So so like say for instance, B positive blood has B specific or beta specific markers that are recognized and rejected by the type of an A positive recipient. Okay, so you've got to be careful and got to have that compatibility to actually go through it. But that's why if you're hearing people talk about like, I'm AB negative, I'm AB positive, I'm O negative, this is what the positive and negative is. It's the presence or absence of these proteins on the surface of your blood cells. Okay. Okay, so we are going to go over the class activity this week when we're going through it. You guys might want to look at it beforehand because it is pretty hefty and you will be turning it in individually as well. But it's going through sex-linked traits with Randy and Sharon Marsh from South Park fame. And basically, you're going to go through, and pretty much all of this, I think, is sex-linked and complex inheritance. So for everything you do on this, it will always have an X and Y, or XX and XY. Okay, so you can still do Punnett squares, but when you do your Punnett squares now, you can automatically write them out like this, right? So we automatically know that we have a 50% chance of having a girl and a 50% chance of having a boy, and then we gotta just add whatever the alleles are onto that. So say we have W and lowercase w, and then we have a uppercase W. Okay, and don't think of this as the curly or wavy hair. That is complex inheritance, but it's not sex-linked. Okay, so sex-linked, not always, but tends to relate to diseases, right? And again, just as an example, I talked about color blindness, and I talked about my mother and father do not have color blindness, right? So I have my dad up here, my mom over here, and let's just say it's a C, right? C is another one, pretty hard to do. So my mom is normal. She is not colorblind. So we don't technically know what her genotype is, right? We would have to either figure it out by doing some DNA analysis, or we can play around with our kids and see what they've got. So we know that my dad is also unaffected. So they've got a good chance, right? But since we know that they produced an offspring that is colorblind, we know that mom's got to be the carrier because the only one that can pass this on is a female because she's the only one with an X to give to that male. Okay, so we've also got a 50% chance that the girl is a carrier. So in my case, I am the daughter of these two, and I have two brothers. One of them is also normal. So I have one colorblind, one normal, which we had a 50% chance, right? Looking at my parents. And look, look at that. We've got one normal and one colorblind. So the odds of me being a carrier are also 50%. So I can do a DNA analysis and I can figure out if I'm a carrier for something like color blindness, or I can chance it and have kids and see if they end up colorblind, right? For me to have a colorblind daughter, so this is now, let's say this is your F1 offspring and I'm included in this. For me to have a colorblind daughter, I would have to reproduce with a colorblind male, okay? Because I would only be able to pass on one of my X alleles. So in order to have the female be an affected person, you would have to have an affected male, okay? But if I have sons and I am a carrier, I've still got a 50% chance of passing it on, right? So this is another one of the reasons why females often do not have these X-linked recessive diseases, especially, is because you've got another X to mask it, okay? So keep that in mind. Also, one more thing that I did not mention, if you see this term autosomes, so an autosome is basically any chromosome that's not a sex chromosome, okay? So that means that we've got 22 pairs of them, okay? There are paired chromosomes except for X and Y. I'm just going to abbreviate it. So except our X and Y. So again, anything other than our sex chromosomes. So if you hear someone referring to autosomes, they're just referring to those other 22 pairs. Okay. I also didn't really talk about polygenic or multifactorial traits. Polygenic and multifactorial, both of them really just mean that, and oftentimes they go together, but these are things like eye color, height, and a few other ones. But polygenic means that it's influenced by multiple genes, okay? So things like height and eye color, those aren't just associated with one gene. It's a little bit more complicated than that, okay? So you can have a trait that's polygenic, or it could be multifactorial, or it could be both. Multifactorial means that it's the genes plus the environment. So you can imagine this plays into height as well, right? We've got multiple genes playing into height that you're receiving from both mom and dad. You're receiving different alleles on each of those genes, but the environment also plays a factor, right? Like I'm a pretty tall person. When I was a kid, constantly everyone was asking me if I drank a lot of milk, right? It's just kind of a joke you say, but those kind of things do factor into how tall or how robust of a human you're going to be. So something like height is both polygenic and multifactorial. Something like, let's say, eye color is polygenic. Probably not really influenced by the environment unless you're putting weird chemicals in your eye, which is arguably not necessarily the environment. But yeah, I forgot to mention those earlier in the lecture as well. So please make sure that you know what polygenic and multifactorial are.